Hello guys, I hope you're all doing well. So this is the, for the first exam, this is the big bulk of your exam. The most important chapter is this one. Out of 50 multiple choice, I don't know, we should have 25 or around 25 from this chapter. So make sure you understand this chapter and you know it. You do need it for the rest of the semester. So anyhow, without any more introduction, let's go ahead. Chapter 5 for the first exam is the most important one. So the molecules of life, and we already briefly, I talked about it in the past, there are 4 macromolecules. There are 4 macromolecules. And these 4 macromolecules are spread into the cells like this. Plant cells are rectangular and animal cells are round. So plant cells would be something like that. You guys are probably having fun with my demonstrations. But anyhow, so plants in general, plant cells are rectangular and animal cells are round. And then we'll talk about what these 4 macromolecules, where they are located in a cell. But before I get to that, we need to get into the macromolecules, the carbohydrates, lipids, proteins, and nucleic acids. These are, I call them building blocks. So throughout the exam or throughout the semester, you might see this. So I call them building blocks. The reason I call them building blocks because they are like the blocks that they make a building. The building is made up of, a wall is made up of chalk or wood, and the window is made up of glass, so on and so forth. But as far as the cell goes, there are 4 building blocks that they are in a cell. Okay, the macromolecules are polymers. So a monomer is, it means 1 glucose molecule and 1 amino acid. It's just a monomer. And polymers, when you have many glucose molecules attached to each other like this. So each one of my finger is a glucose molecule. And when they attach to each other, it's called a polymer. So polymers is a long molecules consisting of many similar building blocks. The repeating units that serve as the building blocks are called monomers. 3 of the 4 classes of the organic molecules are polymers, and those 3 of 4 are carbohydrates, proteins, and nucleic acid. So fats are really not that kind of a building block. They are not, I'm sorry, they are a building block, but they are not polymers. You will see it. So the synthesis and breakdown of polymers are possible by enzymes. That's something I will mention. I will talk about it later on throughout the semester, possibly not this exam. I think it's exam number 2. Enzymes come up in exam number 2. So the dehydration, it means when you remove, or condensation occurs when 2 molecules, 2 monomers bond together. Okay, when you have 2 monomers come together, and of course there is 1 molecule of water leaves the combination of these 2, and that is called dehydration. Remove or condensation, remove of loss of 1 water molecule, remove of 1 water molecule. And then hydrolysis is when you have a molecule, a polymer, if you would, more than 1 molecule, 2 glucose molecule or amino acid. When you break them down, you do need a molecule of water. Okay, so when you break these 2 molecules apart, you need to add a water molecule to it, and that's called hydrolysis, reaction that break down water. Here they are. They're showing it to you. This is a polymer. A, B, C, D, 1, 2, 3, 4, whatever it is. One end is a hydroxyl, and the other end is just a hydrogen. When these 2 come together, right here, and this one come together, the OH of this one is combined with the hydrogen of that one. It gives you a water molecule. Okay, So this number 4 is attached to number 3. And opposite of that, when you want to break it up, then you have to add water molecule here, and then this molecule will get OH, and that molecule will get H. So it breaks apart, and that's called hydrolysis. Lysis at the end of the word in your prefix suffixes, it means breakdown. Okay. Polymers, let's see what this animation is.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydro.
Okay, I'd like to stop that. This, and you saw the blue one on top, these are enzymes. Okay. So you saw another one from top was coming, the blue one or the purple one from top was coming, putting them together, and you have one from the bottom. Right now it's breaking them apart. Those are enzymes. Okay, so let's see.
Release enzyme.
Okay, great. Here is the very first molecule we are going to talk about, carbohydrates. I didn't label them for you, shame on me. So from here on, it starts carbohydrates. Okay, so the carbohydrates, this is a glucose molecule, which has 6 carbon. One carbon number, I should put my glasses on, and then carbon number 1, carbon number 2, number 3, number 4, number 5, number 6. So the carbons are numbered. And this molecule, in nature, rarely you see it straight. Most of the time in nature, most all of the time in nature, it's a ring. So the ring in here is 6-sided. That's what we call it, hexagon ring. And then, but carbon number 1 starts from here, carbon number 2, number 3, number 4, number 5, and number 6 is up here, okay? And then at this corner is the oxygen. If you look at this molecule and you pay attention, all of the carbons have 4 covalent bonds. Hydrogen has 2 covalent bonds. Oxygen has 2 covalent bonds, one with this hydrogen and one with this carbon, and so on and so forth. So we are following the laws of the valence, the covalent bond, the valence. Remember, they have 4, oxygen has 2, carbon has 4, nitrogen has 3, and hydrogen has 1. So we are following all of those rules in here. And so during exam, you should be familiar with this molecule. I'll show you a molecule like this, and I ask you, what is the name of that molecule? You should recognize that as a monosaccharide. Mono, it means 1, saccharide, it means sugar. Not necessarily you have to say it's a glucose molecule. If you say it's a glucose molecule, you're right in this case. But if the hydrogens, hydrogen and oxygen is upside down, which we'll see it is galactose, it's not a glucose, then you lose points because you're not right. And then, of course, this is the glucose molecule again. They're showing you the hexagon side. Usually your textbook show this format for the rest of the semester. So a disaccharide is formed when a dehydration reaction joining 2 monosaccharides together. This covalent bond between the 2 is called glycosidic linkage. Okay, very important. You should know that one, glycosidic linkage. I'll refer to it a few times later on here in a minute. Okay, so here are monosaccharides, and then if you would, this is a hexagon, of course, of a glucose molecule. Look, where the OH is. This is what I was talking about. On carbon number 4, the OH is on the bottom. And of course, on the top, you have hydrogen. On galactose, which is found in milk, okay, that is found in milk, and I will talk about that later on. Combination of lactose and glucose is in the milk. That's a disaccharide. But just galactose by itself, look at carbon number 4. Carbon number 4, the OH is on top. And then, of course, you do have fructose. Fructose is a pentagon, and it has 5 sides. It's not a hexagon. It has 5 sides. And then you have 1 carbon on this side, and then 1 carbon on that side, and it's 5-sided molecule. We call it pentagon. We have another molecule which is 5-sided, is pentose, which you will see it later on. Here it is, the dehydration. So when you combine these 2 sugar molecules together, you remove the water molecule. You already talked about that. And then between these 2 glucose molecule, between that glucose molecule and that glucose molecule, you have an oxygen. The covalent bond here, right, you're seeing it, the covalent bond here is called what? Glycosidic linkage. Very good. So where should I write it down? Glycosidic linkage, okay? So that's a glycosidic linkage. 2 glucose molecules are together, combined together. The name of the molecule, the name of the disaccharide is called maltose. These 2 together, a glucose and a fructose combined together, the name of the molecule is sucrose. Okay, glucose plus fructose, it gives you sucrose. So let's see this animation also.
Sometimes organisms link sugar molecules in pairs to form disaccharides. Here are several examples. Plants make sucrose by joining glucose and fructose. Sucrose circulates in plant sap, and we obtain it from sugarcane and sugar beets and use it as table sugar. Lactose is formed by joining galactose and glucose. Lactose is the disaccharide that gives milk its sweet taste. Maltose consists of 2 linked glucose molecules. Digestion of starch in a sprouting seed or in the intestine of an animal produces this disaccharide.
So make sure you know those. Make sure you know where you find lactose. And lactose is made up of galactose and glucose. Sucrose is found in cane sugar, the sugar that you put, not the NutraSweet, not those sorbitols, whatever it is on the market, but the natural sugar that you put in your coffee, that is a sucrose, the natural sugar from sugar cane, not those synthetic sweeteners. I'm not talking about those, they're different. And then finally, maltose is just combination of 2 sugar molecules, 2 glucose molecules together is called maltose. So these are, you already know 3 monosaccharides. I talked about it. And now you do know 3 disaccharides. Okay, di it means 2, and saccharide it means sugar. Okay, oligosaccharides and polysaccharides. Disaccharides is 2, of course, you know that. Then oligosaccharides, when you have a sugar molecule, it has monomers, 3 to 10 monomers are attached together. It's called oligosaccharides. Oligo, it means a few. It is in your prefixes and suffixes, and you will see that. You will see this term later on, and when we talk about cell membrane, you will see it. And then polysaccharides, from number 11 to infinity, it is, God knows, there is no number, like starch, glycogen is a polysaccharide, okay? The architecture and function of polysaccharides are determined by its sugar monomers and positions of the glycosidic linkage. Definitely, they are alpha linkage, and I will talk about it. Glycosidic linkage, there are 2 types of them, alpha and beta. So here is a sugar molecule that is found in potato, starch. You should know that starch is either straight or branched, okay? So starch molecules in potato or whatever you eat in rice, what happen? what have you, it is either, they are either straight or they can be branched like here or branched like here. So that's starch molecule. On the other hand, well, glycogen is found, they mention it in here somewhere, it should be in my PowerPoint. Glycogen, which is in animals, starch, they call it animal starch, that is always branched. So glycogen is never, right here. Glycogen is never straight in our cells, the cells of our body. Glycogen is always starch, glycogen is always branched, and starch is either branched or straight. And starch is found in plants products, and glycogen is animal sugar, is animal polysaccharides. Okay, so let's see what this animation says. There's a lot of animation in this chapter.
Polysaccharides are polymers, long chains consisting of hundreds to thousands of linked monosaccharides.
Okay, great. And cellulose, okay, in this diagram, he didn't talk about it, but that's okay. You have starch, as I said, it is always straight. Glycogen is always branched. They didn't show you guys here that glycogen is branched. And then of course, cellulose, we need to talk about that. Look at the bonds here. There's a difference between the bonds, the linkage, the glycosidic linkage of the bonding between the sugar molecules. This one is look like it's upside down, okay? But these are all straight, and then that makes it. So quickly, this is a beta linkage, and this is alpha linkage. This is also alpha. Okay? So let's go the difference between alpha and beta. The alpha, as I said, it doesn't show it. Okay, well, I need a disaccharide. So remember, on carbon number one, right here, if the OH is on top, the OH on top, it is a beta molecule. But the OH is on the bottom right here, it's an alpha molecule, okay? So does it make any difference? Yes, it makes a huge difference how the glucose molecules bind to each other. So the OH of this molecule, the OH of this molecule combines with the H of this molecule and form this one right here. Okay, however, in beta glucose, in a beta linkage, what happens, the OH of this one is combined with the H of that one, and it gives you the beta linkage. So that's the difference. Plants make them both, okay? We animal cannot, we human, let's start with human, cannot break down beta. So cellulose, this would be cellulose. So when you eat celery, celery has a lot of this. When you eat fiber, food, fruits, vegetables, they have a lot of cellulose in the cell wall of the cells, and then we cannot break them down. We do not have the enzyme called cellulase to break down cellulose. So we eat fiber in order to cleanse out our digestive tract and so on and so forth. However, when we eat starch from plants that are alpha glucose, they have alpha linkage, as I talked about it before, we have the enzymes to break that down. Okay, we do have the enzyme to break this down. But we do not have the enzyme to break this beta linkage down. Anyhow, I think I said enough. Glycogen here again, they're showing you it is branched, and these molecules. Structure of polysaccharides, the polysaccharides, cellulose is a major component of the tough wall of plant cells, as I talked about it. Like starch, cellulose is a polymer of glucose, but the glycosidic linkage is different. And it is, the difference between is based on two ring forms for glucose, alpha and beta, which I talked about it. Let's see, this is not animation. Sorry, I thought this is animation. Here is a cell wall, and these are the green dots are chloroplasts. The cell wall is made up of cellulose, which we talked about. And we human do not have cellulase. Ace at the end of the word, it means enzyme. We humans do not have cellulase. Ruminant animals like cow, sheep, goat, they do have cellulase, but again, there are microorganisms in the gut that makes this enzyme so they can eat grass and digest it and break it down and to single sugar molecules, but we do not have the enzyme cellulase. Okay, again, I think everything is, the straight chain of sugar molecules, glucose molecules, can turn into alpha glucose, which I use carbon number 1, the OH is on the bottom, or beta glucose, which OH is on the top. I'm beating the dead horse. Right here is the alpha and beta. This is the starch linkage of 1 to 4, 1 to 4 linkage, which is, as you know, 1 to 4. The linkage here, 1 to 4, because this molecule or the OH is on top on the beta, and that's why you have a beta linkage on these molecules. Okay. So starch alpha configuration is largely helical. It means like a helix, twisted. And then beta cellulose configuration are straight usually, and they are not branched, okay? So some hydroxyl groups are on the monomers of cellulose can hydrogen bond with hydroxyl of the parallel cellulose molecules. So it gives them strong fiber. Another carbohydrate molecule, which is unusual, well, we don't know where to stuck them. They put them in with carbohydrates. It's chitin, and you will see it when you take zoology. You will see it in, and then of course in here you will see it in fungi. Again, we will not talk about them too much. We do not talk about organismal biology here that much. Individual organisms, just mostly molecular biology. So chitin is a polysaccharide, is found in the exoskeleton of arthropods, and it has a nitrogen. And I'll show you a picture of it. Chitin is also support of cells in many fungi. Yeah, 2 places that is common in nature. You see them a lot. They're all over, but these are the 2 places that you're familiar with. If I say eggs of nematodes, you probably do not know what I'm talking about, but when you take zoology, you'll say it's found in eggs of nematodes. Anyhow, here they are. They use chitin to make strings, flexible rocks, and then so on and so forth. And right here, what I'm concerned with, that you recognize, this is a glucose molecule, right? A ring format, and here's a nitrogen. Okay, it has a nitrogen with 3 covalent bonds. Right here, 1, 2, and 3. 1, 2, and 3 covalent bonds attached to the nitrogen. Okay, let's move on. Lipids. So the next macromolecule, it's not a polymer, it's lipids. Lipids are, let me see how much I talked. Lipids, I will talk another 15, 20 minutes. Let's finish lipids and then take a break. And as I said, let's break this chapter to 2 sections. Lipids are a diverse group of hydrophobic molecules. As you know what hydrophobic means, it means they do not like water molecules. They do not mix with water molecules. Lipids are the one class of large biological molecules that does not include true polymers. We talked about that. I said that. The unifying features of lipids is they mix poorly, if at all, with water. I already talked about that. Lipids are hydrophobic because they consist mostly of hydrocarbons, which form nonpolar covalent bonds. Most biologically important lipids are fats, phospholipids, and steroids. Those are the ones. Waxes, for our purposes in this class, we will not talk about them. We see some waxes in arthropods, in human dough, wax ear, so on and so forth. But we do not talk about it in here too much. Okay. So they call them fatty acids because on one end, they have an acid group. You remember this? We talked about this, an acid group. And the other end, they have a methyl end. So you remember this one. At the beginning of semester, I draw a methyl for you like this. Okay, so that's a methyl end, hydrogen here. So with carbon in the center, and you have 4 hydrogen attached to it on the side. So, but this hydrogen right here is gone, and this carbon from the acid is attached to it. But this is not really a fatty acid. That's what fatty acids are made up of. Between these 2 carbons, between this carbon and that carbon, you guys, there are many carbons. Usually, the number of the carbons in fatty acids are even numbers. There are a few fatty acids that they are odd numbers, but there are not too many of them in nature. So here it is. Here is the methyl end. Here is the acid end. And here are the carbons. If all of the carbons are attached to, all of these carbons are attached to the hydrogen right here, so they call this a saturated fatty acid. It means all of the carbons are saturated. They're full of, and you know what saturated means. They're full of hydrogen. However, like hockey players in old days, you see they're missing a few hydrogen, they're missing a few teeth, then these molecule is called unsaturated fatty acid. And remember, covalent bond, carbon must have 4 covalent bond around it. So here is 1, 2, 3, and 4. Okay, so 4 covalent bond. If they lose the hydrogen, then between the 2 carbons must have a covalent bond. Okay, so if they use a hydrogen here, this hydrogen is gone. There should be a covalent bond right here. So anyhow, my drawing board, I hope I'm making some sense. So this molecule is called unsaturated fatty acid, and this molecule is called saturated. This top molecule is called saturated fatty acid. Okay, another molecule which is very important is glycerol. Glycerol has 3 carbon, 3 OH, okay, and it has 5, yes. It has 5 right here, hydrogen attached to it, to these 3 carbons. All of these carbons have 4 covalent bonds. Look at it. And then, of course, oxygen has 2 covalent bonds, 1, 2. So that's a glycerol molecule. What happens, you will see next few slides, that if you have a chain of fatty acid attached to this glycerol molecule, then it's called monoglyceride. Okay, but if there are 2 fatty acids attached to this glycerol molecule, you will see it. It's called diglyceride. 3 of them is called triglyceride. 90% of the fat we eat in our diet are triglyceride. 90% of the fat we eat in our diet are triglyceride. And of course, you know, 1 gram of fat gives you 9 calories. Okay, here we go. So this is the fatty acid. This is the methyl end. This is the acid end. The OH of the acid end is combined with the H of the glycerol, gives you water molecule. So when this molecule, this fatty acid, when this fatty acid attaches to this glycerol, the name of the molecule is monoglyceride. Okay, the 3 fatty acids here, they should not be same length. It can be varied. This length of them. One of them has 8 carbon, the other one has 6 carbon, and the other one has 10 carbon. Doesn't matter. Of the saturations, it doesn't matter. One of them could be have unsaturated fatty acid, the other one has saturated fatty acid. It just doesn't matter. Okay. Oh, one other very important thing, I'm glad it came up, is the ester linkage. Right here, the bond, the covalent bond, remember the alpha, the glycosidic linkage is a covalent bond. Ester linkage is also a covalent bond right here. That is an ester linkage right here. The linkage between a glycerol and a fatty acid. Okay. So here it is, combination of water, a combination of fatty acid to glycerol gives you that. And then animation.
Butter, lard, margarine, and salad oil are composed of lipids called fats. make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats like butter and lard are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
Okay, I'll give some nutrition tips at the end also. Okay, here are as the linkage. I talked about it. Let's move on. And of course, he's talking about the saturated fat like butter or lard. They are at room temperature. They are all saturated right here. Okay, but unsaturated fat right here, they're liquid olive oil, vegetable oil that you use for your cooking. Okay, fat made from saturated fatty acids are called saturated fats and are solid at room temperature. Of course, most animal fats are saturated fats. Fats made from unsaturated fatty acids are called unsaturated fat or oils which are liquid at room temperature. Plant fats and fish fats are usually unsaturated fat. Okay, the diet rich in saturated fats, again, nutrition tips contribute to cardiovascular disease. Eating hamburger, hot dog a lot once in a while is okay, but eating them a lot, of course, it leads to heart disease, they clog up our capillaries, atherosclerosis. But anyhow, hydrogenation is the process of converting unsaturated fats to saturated fats by adding hydrogen molecules. That's called the process of hydrogenation. Hydrogenation, vegetable oil also creates unsaturated fats with trans double bonds. These trans may contribute more to saturated fats to cardiovascular disease also. Okay. Certain unsaturated fats aids in synthesis in the human body. These must be supplied in diets. These essential fatty acids include omega-3 and omega-6. He didn't talk about omega-6, but that's okay. Maybe it comes up, which are required for normal growth and our neurons, the cells in our brain, in the nerve cells in our body, they are protected by omega, the fats around them is by omega-3. That's why it's recommended omega-3, omega-6. You should have it in your diet. That's why scientists recommend eating fat. And the diet that does not have any fat is not recommended. What are functions of some fats? Okay, great, good questions. The major functions of fats are energy storage. We talked about that. I already said that. It has more energy than any other macromolecules, 9. Humans and other mammals store their long-term food reserves in adipose cells, adipocytes. Okay, sites at the end of the cell, CYTES in the cell. Adipose tissues also cushions vital organs and also insulations to keep the animals warm. What is the structure of phospholipids? Okay, that's another very important molecule that we need to talk about. Phospholipids are 2 fatty acids and a phosphate group are attached into glycerol. You see some pictures of it, 2 fatty acids, tails. Remember, if this portion of my hand right here is a glycerol, 3 fatty acids attached to it, right? Now, what this next diagram you will see, instead of this one, it would be a phosphate group, which is choline attached to it, which you will see it hopefully, and that would be a fatty acid right here. So here is a glycerol molecule that you saw it before, and attached to the carbon, this carbon is a phosphate group. That's what they call them, phospholipid. And then you have a choline group, which has a nitrogen. They call it choline group. And then the other two carbons, you remember glycerol had 3 carbons? This carbon, a fatty acid, it could be kinked, they call it kinked. It could be unsaturated fatty acid, and this one saturated fatty acid. And they are, they call this the head of the molecule. Right here is called hydrophilic head. It means the head likes water. And these 2 fatty acids here, they do not like water. These 2 are hydrophobic tail. Okay, so in a cell membrane, you have 2 layers of these guys. The heads are facing inside of the cell, full of water. Outside of the cell is also water. The heads are facing outside of the cell. So outside and inside of the cell, they both are full of water, and the heads are facing outside, the heads are facing outside. So the cell membrane is 2 layers. That's what they call it, bi-layer. It's a phospholipid bi-layer. So the phospholipids are 2 layers of them, and then the tails are facing each other, just like an Oreo cookie. You have the chocolate on top, chocolate on the bottom, and in between is all that creamy, gooey stuff, which is good for your heart. No, I'm just kidding. It's not good for your heart. Okay, so we talked about phospholipids, very important. You should know them. And so you will come back to this next exam material. But right now, you should know it. I'm not going to spend, for exam number 2, I'm not going to spend much talking about the structure of phospholipids. We talk about the properties of it later on, but not the structure anymore. Okay, steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. Cholesterol is the mother of all of the steroids. Is a type of steroid, is a component in animal cell membrane and precursor for which other steroids are synthesized. So they're precursor to other steroid molecules like testosterone, estrogen, progesterone. They all come from cholesterol. Okay, a high level of cholesterol in the body may contribute to cardiovascular disease, of course. Here is a molecule of cholesterol, as you saw it. Again, if I break these bonds, and this can become a straight long chain. You might ask, why, Amir? Why they put steroids with fat? Well, because if you break these bonds, all of these, you break them, it becomes a long chain of carbon. And that's what carbohydrates are. But they do form a ring, okay? Unlike polysaccharides, they form a ring, and between each ring, there was a glycosidic linkage. But here, there is no glycosidic linkage between that ring and that ring. Between this ring and this ring, there is no glycosidic linkage. They're like houses in San Francisco. They're right next to each other. But the houses in Stockton, they have a yard between them, a little bit of yard, or whatever amount, I mean, 5 feet, 10 feet, whatever it is. And the houses in Stockton would be the polysaccharides. The houses in San Francisco, they're right next to each other. They are steroids. They are cholesterol. Okay. Proteins, let's stop here. Include a diversity of structures. We come back to all of these molecules throughout the semester. You should know them now in great detail. So later on, we should not have much problem. All right, I'm going to stop.